Welcome on this video. Today I will talk about viruses. Viruses are the smallest type of infectious particle. This means that compared to a bacteria, for example, viruses are much smaller. A typical size of a virus is about 100 nanometer in diameter, but it can range between 10 nanometer to about 300. So viruses are tiny, very tiny. Scientifically, the viruses are not living, but a complex collection of organic matter that is able to self-replicate. They mostly consist of a shell made of protein, which inside contain either DNA or RNA, with enzyme for replication and manipulation of their genetic materials. The structure of virus differs between different types of viruses, but as a general rule, they contain a capsid, which are made up of capsimer proteins. The capsid is important because inside this capsid we can find a genetic material, a strand of genetic information, usually DNA or RNA, which can be single or double-stranded. The capsid can be either icosahedron shape or a helical shape. This is the virus genome. It uses this with the help of various molecular machines to replicate itself. Some viruses also have envelope usually consisting of a lipid bilayer that carry the capsid in genetic material. But again, some viruses don't have it, and they only have the capsid itself. And this is the spike. It's a glycoproteins, and we can have a hemagglutinin. It's a esterase. We can call the latter two the K because they are receptors. Let's look more deeply and let's join the virus journey, reproduction and replication. As you know, viruses have their own genetic code, but they don't have the machinery to copy their DNA. So viruses need your cell's machinery to reproduce themselves. Cells are the smallest unit of life. They manufacture protein, replicate DNA, and store resources. This makes them perfect hosts of virus, armed with its genetic material. Viruses are able to infect healthy cells and convert them into mindless virus production robots. The virus uses this other protein code covered in small molecular receptors to bind and join to the membrane of a cell. This is essential, as it is what determines which cell a virus is able to infect. Once joined, the virus uses several complicated biological pathways in order to force the cell to either accept its genetic material or take in the entire virus itself. Once in the cell, the virus will then begin to use its molecular arsenal to hijack the cell proteins manufacturing system and DNA replication mechanism. After the virus integrated itself into the cell's own DNA, the cell will then replicate its DNA and use to manufacture protein without the cell even knowing. It produces viral protein shells, filling them with virus DNA, effectively making copies of the virus and assembling them. The virus-making machine begins to take over more and more of cell's protein, manufacturing ribosomes, shooting out to thousands of new viruses which quickly fill the cell. These viruses then cause tension within the cell and by several different means burst from the cell with the intention of infecting new hosts for the same purpose. Okay, that's all for the journey of viruses. Let's meet in another video for a new topic.